Assalamualaikum everyone. Welcome in today's session. Sorry for back noise. Uh, I would like to Miss Hanfa for today's session. Miss Hanfa, kindly floor for you. Please continue.
Okay, sir. Assalamualaikum everyone. Welcome to today's seminar on Mega 11, a powerful and widely used software in bioinformatics for molecular evolutionary genetics analysis. So, Mega 11 stands for molecular genetic analysis, and we use the version 11 that originally developed in 1990 by Kumar. The aim of this session is to provide a comprehensive overview of Mega 11, including its function, importance in research, how to install, use it, how to integrate it with other related tools such as ITool, GSDS. Mega 11 is designed to simplify complex analysis for a user without programming knowledge. It offers researcher a friendly interface to perform such tasks such as sequence alignment, phylogenetic tree construction, molecular studies. For example, if a virologist uses Mega 11 to study mutational pattern of COVID-19 by aligning viral genome and construct phylogenetic tree to understand its divergence. Next slide. What is Mega 11? Mega 11 is complete cross-platform software suite that is designed for bioinformatics research in the field of biology, genomics, and systematics. It allows us to align DNA, RNA, any protein sequence, reconstruct a tree, and estimate parameters like divergence time and genetic distance. It may also include as non-coding region, wide algorithms support like neighbor joining, maximum likelihood for phylogeny, muscles, cluster W for alignment, and also use data format compatibility like FASTA, Mega, and for example, if a student working with 16S rRNA gene from different bacterial species, Mega 11 allow us to align those sequences and construct a tree and analyze their divergence allow within a one platform without any description. Am I audible?
Yes, you are audible. Continue.
Okay, next slide. In this slide, we will discuss about role and importance in research. Mega 11 is deeply integrated into many biological disciplines. It supports three primary applications like phylogenetic analysis, sequence alignment, and molecular evaluation. So first of all, we discuss phylogenetic analysis, in which help us to reconstruct evolutionary tree to understand genes duplication or functional divergence. Sequence alignment, in which detect homologous or conserved region revealing critical domains in proteins or genes. And molecular evaluation, in which we discuss about allow researchers to estimate uh, study about selective pressure and calculate pairwise genetic distance. For example, a Mega11 has been used in conservation genetics to analyze mitochondrial DNA of any species and understand population divergence. So it is also applied in oncogenomics, like comparing BRCA1, like BRCA1 is the gene of breast cancer mutation across the species. Next slide. Why we use Mega 11? So researchers choose Mega 11 for its ease of use, comprehensive set of tools, usually friendly user and free available, rich documentation and sports, and also help for small and medium dataset. So Mega 11 is also ideal for student learning molecular phylogenetics, labs comparing viral or bacterial gene families, researcher analyze multiple protein isoforms. For example, a researcher working on chloroplast gene across the different angiosperm using Mega 11 to analyze the sequence, build trees, and publish high confidence phylogenies without need to install multiple tools. It may also help as active development continuously updated with new features, algorithms. Next slide. Am I audible? How to
Yes,
download and
yes,
install
continue.
Mega 11. Okay. So in this slide, we will discuss about how to download and install Mega 11. So first of all, installation of Mega 11 is very easy. Go to the official website www.com Mega Software. Click the download tab, choose our OS version like Windows or Linux, download the installer and run the install and follow the instruction. Accept the license and choose the install path. The software is very lightweight and installed in under 5 minutes. In this slide, you will see that Mega 11. Next slide. After Mega 11 download, we will first of all discuss about performing a multiple sequence alignment. Performing MSA. What is MSA? Multiple sequence alignment. MSc is the one of the most fundamental step in bioinformatics. It involves aligning three or more sequence, either DNA, RNA, or a protein sequence, to identify region of similarities. They may also tell us about the conserved region that indicates structure domains, functional motif, or relationship between the species. MSA is not just about the matching sequence, it is also about to understand how biological information is conserved or diverged across the organism or a gene families. In Mega 11, MSA algorithms are provided each designed to handle different type of dataset. First of all, we will discuss about muscle. Muscle, which is stand for multiple sequence comparison by log expectation. It is very well known for its speed, accuracy, and especially suitable for the large databases. Basically, we use muscle for large databases. It also used to refine techniques to improve the quality of alignment and is generally default choice is many workflows. For example, when we align hundred of viral protein sequence, muscles help us to complete our task easily and making it ideal for outbreak studies like tracking COVID-19 variants. Second thing is the second algorithm is cluster W. Cluster W is traditionally and widely used for alignment method. It is also progressive alignment strategy, starting with the pairwise alignment to build a guide tree and then align sequence based on their order in a tree. Cluster W is especially useful when the number of sequence is moderate like 20 to 30 sequence, and they also choose such as comparing different isoform of the same gene in a human. The third algorithm is MAFT, which is stand for multiple sequence alignment using for fast transform, that is recognized for its high accuracy, particularly with high divergent sequence or with the dealing with long genomic regions. It is very efficient when you have a sequence from different species such as plant, animal, or any species as aligning hemoglobin protein sequence across the vertebrates or invertebrates. So if we perform in MSA in Mega 11, so first of all, we open Mega 11 and go to align, build, and select align, in which the bar show build and edit and build alignment. Then import our sequence, which is mostly in FASTA format, which is standard format for each sequence is labeled and presented is clean and readable structure. Once the sequence are loaded, select your desired alignment algorithm, for example, muscle cluster W. If muscle is selected, if then their speed and are very important. Click align, then calculate mega with process the alignment and display the results. It is important to manually review the alignment and check for misaligned region, gap, or unexpected matches. So finally save the aligned sequence in mega format, which is mega internal format required for building the phylogenetic tree, which is discussed next. For example, if we have 500 spike protein sequence from different COVID sample around overall the world by aligning with these muscle, we can detect conserved domains, mutation hotspot or insertion or deletion, all critical values or changes in vaccine or drugs in drug designing. Next slide. This page will be show you that how to align and how to align MSA with muscle or a cluster W. This is the sequence of a species that is pasted here. Next, in the next slide, we will discuss about the construction of phylogenetic tree in Mega 11. So, after the MSA through cluster W or a muscle, we will construct the phylogenetic tree in Mega 11. So, the next logical step is to refer the evolutionary relationship among these phylogenetic tree construction. A phylogenetic tree is basically is a branch diagram that show how a different organism or a gene are related to each other through evolution. Like these trees are central to many biological studies from tracking disease outbreak to understanding gene families. In next slide. In the phylogenetic tree, we will see that node that represent the taxa, like gene or a species or their common ancestors. Branches that show the connect between nodes and represent evolutionary lineage. Longer branches typically imply greater evolutionary changes or divergence. And then discuss about root. Of the tree is the most recent common ancestor of all sequence that is connected or being studied. A clade is a group of a sequence that from a common ancestor think it is like as a family within a tree. In Mega 11 offers three methods for tree constructions. So first of all we will discuss about neighbor joining. So what is neighbor joining? Neighbor joining is a distance-based method. It calculates pairwise genetic distance and then construct a tree that minimizes the total branch length. It is very fast, efficient, and suitable for large databases such as hundreds of bacterial 16S rRNA sequences. So next next method is maximum likelihood. Maximum likelihood is used as statistical model to estimate the tree that is most likely to explain the observed data. It is more accurate than neighbor joining, but it requires more computational time. So maximum likelihood is best when high confidence and model accuracy are required. For example, in reconstruct the evolution of viral surface protein. And the next step is maximum parsimony that find the tree with the fewest evolutionary changes. It is the simple method based on the principle that can become less reliable as dataset size or complexity increases. To build a tree in Mega 11, first of all, we upload our aligned sequence we will save in the Mega format. Recently uses in MSA and save the Mega format. Go to Phylogeny in the Mega 11 and construct the tree and choose our method that is depend our research like neighbor joining or a multiple maximum likelihood. So the next step is select an evolutionary model that depend on our research and set the parameter that where the data is nucleotide, DNA or RNA or a protein base. And the next step is enable bootstrap analysis. Say like 1,000 replicates, it add statistical reports value to your branches, helping you judge how reliable each group is. So what is bootstrapping? Bootstrapping in phylogenetic tree like 100 replication, 1,000 replication, it purpose how to reliable the evaluationary relationship within a tree. Branches are labeled with the percentage of bootstrap value, showing how often that grouping occur across all the tree. If a high bootstrap value greater than 70 is suggests that a reliable grouping. After that, we will click the commute to generate the tree. For example, if we are working with the cytochrome C protein sequence, with different 10 different members by using maximum likelihood method in omega 11 we will observe that human or chimpanzee sequence group tightly together confirm that there are close evolutionary relationship like Rodents like mice from different clades reflect that deeper divergence. Next slide. This picture shows that the phylogenetic tree, like root nodes, clade, roots of all the phylogenetic tree. Why we use multiple sequence alignment before the genetic tree to construct in Mega 11? It is because Mega 11 built tree by comparing homologous position in DNA, RNA sequence or a protein sequence. For that comparison, it might helpful the same functional or structural region must be aligned across all the sequence. Without alignment, Mega cannot recognize which nucleotide or amino acid position in one sequence correspond to another. It might may be causing mistake. If in the sequence of DNA or protein have deletion or insertion heading, then MSA detect the gap and align the sequence accordingly. So after that MSA we use to construct the phylogenetic tree that is look like this image. Next slide. In this slide we will discuss about the ITool. While Mega 11 is powerful for analysis and tree building, but it has limited option for visualization and annotation. That's where iTool is used. iTool is an online tool available at itool.embl.de, specially designed to visualize, annotate, and manage phylogenetic trees. iTool for us we can zoom in or zoom out, rotate and rearrange the tree like the tree circular, rectangular and unrooted layouts. It may also mindful for annotation, add custom label, colored branches, symbol or charts to highlight the metadata such as expression level, host species or a resistance gene. It might also helpful for external database, for example, labor charts, heat map showing antibiotic resistance, gene expression, or geographical location. Our iTool allow us to export the final annotated tree in high resolution in PNG form, SVG, or PDF format, which are useful for our publication. So discuss about iTool, how to use it. In Mega 11, that we construct a tree. After that, export our tree as an UV file and go to iTool and upload the file. After the upload the file. Use the graphical user interface to annotate branches, add color, label, or even a pie charts. It all depend on our visualization or representation of a tree. After preview and export our tree for our publication or a presentation. For example, if we have constructed a phylogenetic tree of influenza virus strain, then we use iTool. We can annotate each branch with host species like human, swine, birds, and different isolation like Asia, Europe. This annotated tree gives us clear insight into a transmission and global separate pattern. ITool is basically a web tool that enhances a phylogenetic tree or also helpful for evolutionary biology, compare genomics, microbe genome studies, and epidemiology, like I discussed recently about the COVID. Next slide. This is the overview of iTool. How to we annotate and add colors and label and also rename the nodes or branches with gene name, accession IDs, or host species or sampling location, mutation types, or a resistance marker. We also add pie chart or other graphs to display such as gene expression percentage or host distribution directly on the tree. For example, each leaf node show that the pie chart representing the proportion of antibiotic resistance. And the next slide is all about GS and DS, gene structured visualization, in which GSDS means gene structure display server. Gene structure display server is a web tool based at gsds.pu.edu. It is used to generate visual representation of gene structure, particularly the arrangement of exons, introns, and UTRs untranslated regions, like Mega 11 or ITool, which is deal with the evolutionary relationship, but GSDS help us to visualize how a gene is organized with the genome. So GSDS is basically two type of sequence. First, the genomic sequence, which is include both coding region or non-coding regions, like including introns, and the other one type of sequence that we input in the GSDS, that is CDS. CDS sequence is coding sequence, which is only contain the exons, the part of the gene that are translated into a protein. So why we use GSDS? To compare gene structure between the species, and does have a gene have same number of exon in a human or a mice. To check that, we use the GS and GSDS survey to analyze alternative splicing by comparing of the same genes to identify features like promoter region 5 and 3 UTRs or a large introns. So, and in the next slide, step to use GSDS. First of all, visit the GSDS website, choose the appropriate input format. As I discussed above, CDS or genomic format, paste the genomic sequence and the CDS sequence of our input boxes of any species and submit the job. GSDS will analyze and output a graphical diagram. The diagram shows that exons are colored as rectangular and introns are as line or arrow between the exons. UTR may be shaded differently or labeled. Like if we pasted a BRCA gene 1 sequence, genomic sequence and CDS sequence for both human and mice. GSDS might be revealed that humans have 24 exons while mice have slightly different exon count and exon length. This is helpful in our identify evolutionary conservation or alternative splicing events. Like exon is a coding region of a gene that remain in the messenger RNA after splicing, that is encoded amino acid into a protein that show in blocked or different colors in GSDS, while introns are non-coding region that is removed during RNA splicing. It may also helpful or play a role in regulation and gene expression control. It show in connecting line between the exons. Why it is matter, like alternative splicing intron allow flexibility. One gene can produce multiple protein, while mutation studies many diseases arise from the exon skipping or intronic mutation after splicing. Gene annotation that help in confirming the exon boundaries when annotate new genomes. So we use GSDS to confirm about these alternating splicing, mutation studies, or gene annotation. So they help us exon count, intron lengths, and UTR positions. And then next slide we will discuss about omega 11. Next slide. In this slide, you will see that GSDS server screenshot, and on the left side you see how to use GSDS. So basically, select the input sequence option, paste genomic and CDS sequence, and submit the sequence to generate the gene structure diagram. In the next slide, yeah, like this. Introns and exons show that in yellow color rectangular shape or a red shape. UTRs also labeled with purple lines, purple shaped rectangular of our input data, or that in the format of CDS or genomic, that give this intron and exon with UTRs, length, and exon count number. Next. So that Mega 11 is not a just tool but a bioinformatic platform that enable researchers to understand evolution at the molecular level, from aligning sequence with the powerful algorithms like muscle cluster W map to construct accurate phylogenetic tree using neighbor joining maximum likelihood. So Mega 11 simplify complex biological analysis into an accessible workflow, even from a beginner in the field. What makes Mega 11 even more valuable in its integration with complementary tools. With the ITool, you can take your tree and convert into an interactive, annotated, and publication quality visualization. This is when we want to highlight specific metadata such as host species, expression data, or a resistance gene. On the other hand, GSDS help to shift the focus to structural biology. That is, offering a way to visualize and compare gene, and especially useful in functional genomics and comparative gene studies. If we talk about the advantages and of Mega 11, it is easy to use with non-coding experience, comprehensive algorithms, multiple sequence alignment, and tree building in a one place. So available in also for a window and free and lightweight, easy to install and run on a standard system. But it have some disadvantages or limitation like limited batch processing. Mega 11 have low lack automation or subscripting option for high throughput jobs. It is low or larger database. If we use multiple sequence with a hundred of sequence, it might not helpful. Or if we talk about iTool advantages, iTool have advantages as integrative, beautiful pre-visualization, have support also rich annotation, pie charts, heat map or color label, etc. and also export in high resolution format. That is best for our journal publication. But there is also a limitation is that require internet access or web page. If we upload a high size or feature that is not free version, it that is the main its limitation, or that causing annotation be time consuming. And if we talk about GSDS advantages, it might be helpful for visualize gene model clearly, include in exons, intron, UTRs, and sport across the species comparison. It is also great for isoform and alternative splicing analysis. But is the main limitation is that if we faster header must be first of all we remove the faster header. No sequence analysis, alignment, or evaluation. That is also a limitation of GSDS and cannot be processed large genome or large genomic data. If we talk about uh, which is one is preferred, Mega 11 or a Mega 12. Mega 11 is ideal for teaching or learning environmental position like student and small researcher projects use Mega 11 usually. And researcher doing basic to intermediate phylogenetic analysis for a small sequence or might be 100 to 1000. If we Mega 12 is preferred when you are working with the large databases like in a thousand of sequences. It is very faster computation of trace using maximum likelihood with bootstrapping. Mega 12 is newer and still be undergoing community testing. Many publication, lab, educational institute continue using Mega 11 because of all core features are still robust and trusted. If we talk about a researcher using 20 viral genome for a evolutionary study, that might use Mega 11. If we talk about a computational biologist processing 5,000 metagenomic context, that might be helpful Mega 12. That helpful in speed improvement. So, in the role of bootstrap analysis in Mega 11 or a Mega 12, it is like bootstrap analysis provide the statistical confidence for each branch in a phylogenetic tree. Mega 11 resample the dataset multiple times, like 1,000 times replicate and calculate the tree and report percentage. A branch with greater than 70 bootstrap sport is typically considered much be reliable. Okay, thank you. Any question? Any question you may ask?
Okay, thank you so much everyone to listen us and for your attention. So here question: Can Mega 11 perform codon-based evolutionary analysis? Can Mega 11 perform codon-based evolutionary analysis? This Sanfa.
I am audible.
Yes, you are.
Sorry, my mic is muted. Okay, I will again tell you. Codon-based analysis in Mega 11 basically focus on nucleotide substitution at the codon level, like rather than simple amino acid, like nucleotide substitution at codon level, like silent or maybe called amino acid mutation. If a student might be use codon based analysis, like codon based, then first of all codon DNA sequence like cds is used, might be aligned in codon wise, like triplet preserving alignment. In the file format is mega or a FASTA format. But first thing is that align the all amino acid sequence using muscle or a cluster W. Then we will use it.